Welcome back to Nature Gut Brain. Can weed killer change behaviour through the gut? What if a chemical designed to kill weeds could also affect behaviour, not by directly attacking the brain, but by changing the microbes living in the gut? That is the unsettling possibility raised by a new study published in Molecular Psychiatry. Researchers gave mice glyphosate, studied their gut microbes and behaviour, and then performed a revealing experiment. They took gut microbes from glyphosate-exposed mice and transferred them into mice that had never been exposed, and one of the behavioural changes travelled with the microbes. Here is what the researchers did, what they found, and what we should and should not conclude. So why would glyphosate affect the gut microbiome? Well, glyphosate is the active ingredient in many widely used weed killers. It kills plants by blocking part of a chemical pathway called the shikimate pathway. Plants need this pathway to produce certain molecules essential for growth. Now, humans and other mammals do not have this pathway. That has often been used to explain why glyphosate should be much less toxic to animals than it is to plants. But there is a catch. Many of the bacteria living in our gut do have the shikimate pathway. So glyphosate may not need to attack animal cells directly to produce an effect. It could potentially change which gut microbes survive, which ones grow, and how they interact with one another. Think of the microbiome as a rainforest, so a disturbance does not necessarily destroy the whole forest, but it might favour some species and suppress others, and change how the ecosystem functions. And gut microbes are not passive passengers, so they help process food, train the immune system, protect the gut lining, and produce chemicals that can communicate with the nervous system and the brain. So an interesting question is, could glyphosate rearrange the gut ecosystem enough to influence behaviour? So here's the experiment. The researchers gave adult male and female mice glyphosate in their drinking water for 7 weeks. There were 4 groups of mice, so one group didn't receive the glyphosate, and this was the control group. The others received 0.5, 5 or 50 milligrams per kilogram of body weight per day. The lowest dose matched the European acceptable daily intake used by the researchers. The other doses were 10 and 100 times higher. An acceptable daily intake is not the amount most people actually consume. It is a regulatory limit intended to represent an amount that could be consumed every day over a lifetime without an appreciable health risk. Now, the researchers examined much more than whether the mice became visibly sick. So they measured body weight, organ size, stress hormones, genes involved in the gut barrier, the composition of the gut microbiome, and gene activity in the amygdala in the brain. So this is a brain region involved in emotions and social behaviour. Now, they also tested how the mice behaved. One experiment involved placing a mouse near 2 others, one it had already met and one unfamiliar to the mouse. Healthy mice will usually spend more time investigating the newcomer. Researchers call this social novelty preference. It is a laboratory measure of social recognition and interest. Now, it's not the same as human friendship or empathy necessarily, or personality, but it can reveal changes in how mice process social information. Now, the groups were relatively small, with roughly 10 or 11 mice of each sex at each dose. So this wasn't an enormous experiment, but it had some interesting results. So what happened? Well, the clearest behavioural effects appeared in the male mice. Males in the control, low dose and medium dose groups showed the expected preference for the unfamiliar mouse. But males receiving the highest dose of glyphosate did not. They no longer showed a clear preference for the new social partner. Now, some males also spent less time in the exposed centre of an open arena. Now, researchers often interpret that as an increased anxiety-like behaviour, because anxious mice tend to remain near the protected edges of the arena. However, glyphosate did not affect the animals in another commonly used anxiety test, so it would be too strong to say glyphosate made the mice anxious. Now, the accurate conclusion is that it changed one measure of anxiety-like behaviour. Females showed fewer clear behavioural effects. Now, those receiving the highest dose moved around less, but the researchers did not find the same strong pattern of altered social behaviour in the females. The female social test was also difficult to interpret because even the control females did not show the expected preference for the unfamiliar mouse. Despite all this, the mice did not appear obviously poisoned. They did not lose more weight, their stress hormone responses did not clearly change, and the researchers did not detect major changes in the gut barrier gene that that they measured. Now, that is part of what makes this study interesting. The effects were subtle and behavioural rather than obvious signs of severe toxicity. So, what happened to the microbiome? Well, glyphosate did not wipe out the gut microbiome. It also did not significantly reduce the total variety of microbes living within each mouse. Instead, it changed the makeup of the community in the microbiome. A simple way to think about this is that the number of instruments in the orchestra did not necessarily decline, but the types of instruments and the balance between them changed. So some groups became louder, others quieter, and the overall composition shifted. There were hints that particular bacteria changed too. So for example, bifidobacterium strains appeared to decline at higher doses, but most changes in individual bacteria were not statistically strong enough to be treated as firm findings. The clearest result was a change in the microbial community as a whole. So the researchers also examined gene activity in the amygdala. Now they found that changes in a small number of genes linked to social behaviour, learning and memory, especially at the highest dose. But there was no major shift across the entire set of genes examined. So at this stage the researchers had shown that glyphosate exposure happened alongside changes in gut microbes, changes in behaviour and some changes in the brain. But that still did not prove the gut microbes caused the behavioural effect. Glyphosate could have affected the brain directly. It could have affected the gut and behaviour through completely separate routes. Or the altered microbiome might have played a causal role. To separate those possibilities, the researchers performed the most important part of the study, the microbiome transfer. The researchers took gut contents from male mice exposed to the highest glyphosate dose and from untreated control mice. They then gave antibiotics to a new group of mice to reduce their existing gut microbes. After that, the mice received repeated transfers of microbes from either the glyphosate-exposed donors or the control donors. The recipient mice themselves were not deliberately given glyphosate, and here is the striking result. Mice receiving microbes from glyphosate-exposed donors also lost their normal preference for the unfamiliar mouse. The social behaviour change had effectively travelled with the gut community. Under these experimental conditions, transferring the glyphosate-altered microbiome was sufficient to reproduce the social impairment. But the anxiety-like behaviour did not transfer. That suggests the different behavioural effects may have involved different biological routes. The social effect appeared to have involved the gut microbiome. The anxiety-related effect may have required direct glyphosate exposure or some other mechanism. Now, the changes in amygdala gene activity also did not appear in the recipient mice, so the researchers still do not know exactly how the gut microbes altered social behaviour. Possible routes include microbial chemicals entering the bloodstream, changes to the immune system, communication through the vagus nerve, or effects on other parts of the nervous system. There is another important detail. By the end of the experiment, the microbial communities of the recipient groups were no longer dramatically different. That could mean a temporary, early change was enough to trigger a longer lasting effect. Or something produced by the microbes rather than the microbes themselves may have been responsible. The transplant experiment strengthens the case for a causal role of the gut ecosystem, but it does not yet reveal the precise microbe, molecule, or pathway involved. So how does this fit with other research? This is not the first animal study to report links between glyphosate, gut microbes, and behaviour. Earlier mouse experiments have found changes in microbial composition alongside anxiety-like or depression-like or altered social behaviour. Other studies have reported gut inflammation or damage after longer exposure to glyphosate-based products, but the evidence has not been completely consistent. Some experiments have found only small or temporary changes to the microbiome, and the results can vary depending on the animal species, the sex, the diet, age, starting microbiome, length of exposure, and dose. It also matters whether researchers use pure glyphosate or a commercial weed killer, so commercial products contain additional ingredients that may affect how toxic the final mixture is. The new study adds something important because microbiome transfer went beyond simply showing that microbial and behavioural changes happened at the same time. It demonstrated that the altered gut community could carry one specific behavioural effect into another animal. That is a stronger form of evidence evidence, but it is still evidence from mice. What this study does not show. Now for the essential reality check. So this study does not demonstrate that ordinary glyphosate exposure causes anxiety, autism, depression, or any other psychiatric condition in humans. The transplant experiment was performed only in male mice. The clearest social effect occurred at 50 mg per kilogram per day, 100 times the European acceptable daily intake cited by the researchers. That does not mean that the lower dose was completely without effects. One measure of anxiety-like behaviour changed at the lowest dose, but the central social behaviour finding and the transplant experiment involved the highest dose. Now, typical human exposure through food is generally lower and occurs under very different conditions. Now, mice are also not miniature humans. They produce chemicals differently. Their microbiomes differ from ours, and mice behavioural tests cannot diagnose human mental illness. A mouse failing to investigate a stranger does not mean that it has a human psychiatric disorder, and the recipient mice were also treated with powerful antibiotics before receiving the transplant. That is useful experimentally, but it creates an artificial gut environment that does not resemble ordinary human exposure. And because the study involved relatively small groups and many different measurements, independent replication will be essential. So this study is a really important warning sign worth investigating, rather than proof of harm in humans. Why it matters. So the deeper importance of this work is how it changes the question we ask about chemical safety. Traditional toxicology often looks for visible damage. Does the animal lose weight? Are its organs damaged? Can it reproduce? Does the chemical cause cancer or death? Those questions remain vital, but what if a chemical changes behaviour by rearranging the microbial ecosystem inside the gut? The chemical may not need to directly damage the brain. It changes the internal ecosystem, and the changed ecosystem alters the animal and its behaviour. The researchers describe this broader perspective as ecological psychiatry, the idea that mental health and behaviour are connected not only to the brain, but also to the environmental chemicals, the food, the microbes, and the ecosystem inside us and around us. Now, that doesn't mean that current safety limits are automatically wrong, but it means that microbiome and behavioural effects may deserve a more serious role in future chemical testing. So the most responsible conclusion is that in mice, glyphosate changed gut microbial communities and behaviour, and when researchers transferred the altered microbes into unexposed mice, a specific social behaviour change travelled with them. That provides experimental evidence that the microbiome can help mediate the behavioural effects of an environmental chemical, at least under these laboratory conditions. We now need replication, longer experiments, more realistic exposure levels, and a much clearer understanding of the actual microbial molecules and pathways involved. But the study leaves us with a bigger lesson. Our bodies are not sealed away from the wider environment. The gut is an ecosystem connected to the food we eat, the water we drink, and the chemicals released into the world around us. So when we test a substance designed to alter living systems, perhaps we should ask not only what it does to our human cells, but what it does to the organisms with which we share our bodies. Thanks for watching. If you found this video useful or interesting, please do me a huge favour and click like and subscribe to the channel. It really does help the channel grow and allows me to keep making these educational videos. And remember, stay wild inside and out. They put weed killer in the water, watch the mice from day to day. Nothing looked too different outside, but something underneath had changed. Then they moved the microbes over to mice that never had the dose, and changed in social behaviour, follow where the microbes go. What happens in the gut, 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 don't stay in the gut, gut, gut. Tiny lives are sending signs, all the way up to the mind. What happens in the gut, 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 don't stay in the gut, gut, gut. Tiny lives are sending signs, all the way up to the mind. And the microbes didn't disappear, but the balance moved around. Some grew louder, some grew quieter, like an orchestra changing sound. And it doesn't prove it happens in people, but the strongest dose was high. But chemicals change our ecosystems, and our ecosystems shape our minds. What happens in the gut, 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 don't stay in the gut, gut, gut. Tiny lives are sending signs, all the way up to the mind. What happens in the gut, 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 don't stay in the gut, gut, gut. Tiny lives are sending signs, all the way up to the mind. Wow.